Okay, myself, Dr. Manjit Panigrahi. I'm a scientist at ICR IVRI in the division of animal genetics. And I have been working in the field of livestock genomics for the last 5 to 6 years. And my favorite topic is population genetics. So today I'm going to give a lecture on Introduction to Population Genetics and its application. I will divide my lecture into two parts. The first part will discuss the basic concepts of population genetics, and in the second, here the first time people come to know that some sort of constant factors or characters that is mainly important for the inheritance. Now, what is a gene? The gene is to genetics what atom is to chemistry. This gene term was coined by Wilhelm Johansen. And if you see the picture here on the right side, this is a picture of a cell, and inside a cell you find a nucleus. Inside that nucleus you have some 23 pairs of chromosomes. Inside that chromosome you have the DNA, it's a highly packed DNA, and inside that DNA you find the genes. So this is the concept and how the genes are packed inside the genome. Now, almost in all living things, you find DNA as the genetic material. Now, what is this DNA? If you want to know the power of the DNA, you come to know that we have somewhere around 65 trillions of cells. And in that 65 trillions of cells, in each cell you have 23 pairs of chromosomes if you are a human being. And inside that, if you just unfold the chromosome and join them from end to end, if you join them, you will reach to sun for around 70 times and come back. And this is the power of the DNA in each and every individual. That's why we used to say that हमारे अंदर ब्रह्मांड समाया हुआ है. Okay. So after knowing the power of the DNA, you need to know the timeline of genetics. Being the professor of genetics, I would like to start from the 1800, the formation of herd books. Then the Mendelian concept came into existence, but most of his work was not known until 1900. It was again rediscovered by Sir Mac, Carl Correns, and Hugo de Vries. So after that, people came to know, and then the term gene and genetics and phenotype, genotype, all these things came into existence. And the bottom line, you will find that this is the concept of breeding concept is going on, and the upper side you will clearly see that it's the concept of molecular genetics. So initially people were thinking that there is an effect of major genes. That means one gene controls one phenotype, but later on Fisher, Halden, Wright, all those pioneers of quantitative geneticists, they said that maybe the traits are controlled in a quantitative nature. It's a quantitative inheritance kind of thing. And after that, the use of computers in identifying the breeding values, the estimation of blobs, all those Henderson models, and we are not going to discuss in detail here. And in the upper side, if you see that somewhere around 1928, the genetic material, DNA is the genetic material, was first time people came to know that by Griffith. Then in 1944 by Avery, MacLeod and McCarthy, they confirmed that DNA is the genetic material. Then the Watson-Crick model, this is well known throughout the world, that the double helix of DNA. In 1953 they got Nobel Prize for that discovery. And then the Sanger sequencing, the genetic codes, all those PCR, polymerase chain reaction, then QTLs, quantitative trait loci concept, then microsatellites, mini satellites, all those markers, and like that the molecular genetics moves on. But towards around 2001, that the genomic concept is being introduced in the breeding concept, and then the G-blob was introduced, or the information of genomics is introduced into the breeding concept, and the breeding value was estimated. And then finally we just the human genome was sequenced, then similarly the cattle genome sequenced, the chicken genome sequenced, and side by side we have developed many chips, the SNP chips that I'm going to discuss later on, for almost all the species. Okay, so this is how it got developed. Now if you come to the cycle of complexity, this is the most important equation in animal genetics. Everybody should know that genotype is alone is not enough. In the environment it is existing, and this is the phenotypic expression of an animal, and that is how it is designed. That means phenotype is equal to genotype plus environment. But we are, being the geneticists, we are more concentrated here in the genotype area. So what is a genotype? If you see here, this is a picture of 2 chromosomes in an individual. I am just elaborating it. Now here you can find that this portion is known as a locus, and in that locus you will find 2 different alleles in an individual. You can get maximum 2 alleles at that particular locus. Now here you can see that this is capital P, this is small p, and it is commonly known as, this is the genotype, capital P, small p, whereas capital P is one of the alleles and small p is another allele. So allele is nothing but the alternative form of a gene. Now, after knowing that, what is then allele frequency, gene or allele frequency we frequently use in animal genetics or population genetics? It is the proportion of one allele relative to all alleles. Now, if you see here, you can find that the frequency of capital A, there are 2 alleles at a particular locus, it is considered as small p. That is the frequency of capital A, and this is the frequency of small a, that is Q. So this is how, and if you add them up, it goes to 1, because there were only 2 alleles. Similarly, there were 3 genotypes. So then from there we can calculate the genotype frequency as well. Okay. So there were 3 genotypes. This is capital AA, the homozygous dominant type, the heterozygous, and the homozygous recessive type. Now here from you can see in this example that there are some 60 individuals of capital AA. That means 60 out of 200. That will give rise to the genotype frequency, that is somewhere around 0.3 or 30%. Now for the heterozygotes, it is somewhere 0.5 or 50%. So in this way you can calculate the genotype frequency. And if you need to calculate the allele frequency, what you have to do, you just add them. This one plus half of this one will give rise to the allele frequency of capital A, which is somewhere around 0.55. And the small a frequency will be 0.45. This is how we used to calculate the gene and genotype frequency at that particular locus. Now, why do we study gene and genotype frequency? Because evolution or the microevolution is nothing but the change in gene frequency or genotype frequency. And here I would like to give the quotation of Theodosius Dobzhansky, who said that nothing in biology makes sense except in the light of evolution. And it's a fantastic quotation. I mean, everything is dependent on the evolutionary processes. Throughout generations, from generations, we have been evolving, and till now, still we are going to evolve into a newer form. I mean, without evolution, there is nothing. And this is how life goes from generation to generation. Okay? Now, if it is so, here you can see that evolution of population, it is nothing but change in allele frequency, as I have already told you. And what are the 5 main agents that cause evolutionary change? That part we have to be very, very clear. Being the genetic students, you need to know that. What were the 5 forces? What are they? The first one is mutation. I mean, it is continuously, it is giving rise to newer variety into the population. Then comes the migration. If you move from one place to another, you will change the gene and genotype frequency. Then comes the non-random mating. Then comes the genetic drift. That means if you, I will discuss it later on, I mean after this slide. Then comes the selection. Okay, so these are the 5 major forces which cause change in the gene and genotype frequency, and our objective of selection, as we all know, that is to change the gene and genotype frequency in the desirable direction. Okay. Now, now coming to the simplest model by Hardy-Weinberg law, what he said, that these were the assumptions. Now in mathematics, as we all know, that we always start from simpler forms. So what they said, that if there is no selection, no mutation, no migration, and the population is a very huge one, and there is random mating, then the gene and genotype frequency remains constant from generation to generation. This was the simplest of all model by Hardy-Weinberg in 1908. They discovered it individually, independently. Okay, but so so you can clearly see in this picture that this is the relationship between the gene and genotype frequency. What will happen in a Hardy-Weinberg model? Okay, so here you can see this is the heterozygote. These are the homozygotes recessive ones, and these are the homozygotes of the dominant one. If it follows a Hardy-Weinberg equilibrium, then you will definitely get this kind of relationship between gene and genotype frequency. Here P is the gene frequency of the dominant allele and Q frequency of the recessive allele. It goes 1 to 0, it's 0 to 1 like that, and this is the genotype frequency. Okay, and this is the relationship. Now what is peculiar in it? Here you can clearly see that the heterozygotes are maximum at intermediate gene frequency around 0.5. Okay, and what is the second point? If you can see here, most of the homozygotes which are recessive in nature, they hide themselves inside the heterozygotes like a carrier. That's why we always say that most copies of the minor alleles are in heterozygotes and not homozygotes, and it hides itself inside the heterozygotes. Okay, now this is the peculiarity of Hardy-Weinberg equilibrium, and I am not going into the details of it. We need to see that whether a particular allele or locus is in Hardy-Weinberg equilibrium or not with the help of chi-square test, and that can be measured with these frequencies. Here again I would like to say that P plus Q is equal to 1, and if it follows the Hardy-Weinberg equilibrium, then the heterozygotes also, this is the frequency of homozygous dominant, frequency of heterozygotes, frequency of homozygous recessive, that will also add up to 1. Clear? So this is the equation of Hardy-Weinberg, but life is not that easy. It does not follow this ideal condition always. Population is not idealized. So what Wright and Fisher, who were the pioneers of quantitative genetics, they said that, well, let us introduce some sort of constraint into it. Constant, if there is constant population size, they have not taken a large population size. If there is constant population size, if the population size is known or constant, then what will happen? Okay. So here I would like to emphasize upon 2 points, that there are 2 processes. Already I have told it, but again in a refined form. One is systematic process. The other one is dispersive process. In the systematic process, that is migration, mutation and selection, you measure the gene frequency in a manner predictable both in amount and in direction. You can know the amount of change, you can know the direction in which it moves. Okay. But whereas in dispersive process, which is applicable to small populations, mostly in small populations it arises, you can measure the amount in terms of variance, but you cannot measure the direction. Say for example, I will explain here, and here the random drift comes into it. So say for example, if you see here, let there be 20 individuals in a population, and if it starts from 0.5, few of them goes randomly and fixed here, and that is known as fixed, it goes to 1, and few of them dies out, it goes to 0. So this is how it got lost, and many of them is moving here and there inside that frequency spectrum. But if you increase the population size, say for example 200 or 2000, it goes on, and here it is quite close to the mean value. And if I have to explain it in terms of an example, let us see that you toss a coin for 10 times, you may get a head by 7 times and tail 3 times, you may get 6 times head, 4 times tail, it varies randomly, okay, if it is a small size. But if I toss that coin for 10,000 times, what will happen? You will go closer to the mean value, means somewhere around 5,000, towards 5,000 you will get the head, and 5,000 you will get the tail. Now this is how it moves, and you can measure the drift by variance, genetic variance, that is PQ by 2n, I am not written here, but each line fluctuates irregularly. This is the phenomenon of a random drift. Whenever there is small population, you are bound to get some sort of random drift that moves from, means it's a random fluctuation of gene frequency. Okay. Now, these are the two important phenomena that takes place in a small population condition. Like founder effect, it occurs when a population is initially established by small number of breeding individuals. What it says, few of the individual goes from one place to another and they start developing their own colony, then that is known as founder effect. Okay. Similarly, bottleneck effect. Here you can see that the population size goes through bottleneck, means some sort of constraint, some sort of hazardous situation, maybe some calamity, and many of the individual dies out. Maybe some asteroid hits the earth and you find few of the individuals survives. And from there, if the population grows on, it is known as bottleneck effect. These are the most important questions, and if you appear CSIR, JRF exam, you will find these kinds of questions. Okay. What is bottleneck effect, what is founder effect, and what is Bulmer effect? Bulmer effect is nothing but the reduction in variance due to these causes, and that is known as Bulmer effect. Now, okay, so that is all about the drift and what is happening. I mean, people were thinking that most of the mutations that arise, those are deleterious in nature, and most are favorite in nature and advantageous in nature as well. But this is the theory proposed by Kimura, who said that most of the variation present within a population is selectively neutral. I mean, those changes or the SNPs or the single nucleotide changes are occurring on the non-genic region or intergenic region. I mean, they are not on the codon region. I mean, they are equally distributed on the genome and they have no effect on the protein. This is the concept by Moto Kimura, and it has revolutionized the whole concept of selection theory. Okay, so here you can see that if a mutation comes in, many a times because it is deleterious, it dies out. But most of the times, maybe some of the times, it goes to one and then it gets fixed there, and that is how it moves. So it's a random process. So the neutral theory helped the randomization. Now, after that, if you see in this picture, the genetic diversity is defined as variations in the genetic composition of individuals within or among species. Now, a mutation, as I have already told you, it introduces genetic variation into the population. Whereas drift, random drift, it always have a tendency to either fix the allele or to lose the allele. So it removes that genetic variation from the population. Okay. And there it creates a diversity inside the population. This is the population, and here you will find the diversity. This is theta, or Watterson's distribution. So theta s is equal to 4 ne mu, and it is quite widely derived. And what is it? I mean, the total amount of diversity whenever there is a neutral model, whenever there is mutation drift balance, you will find this much amount of diversity in the population. And here you can see the N is nothing but the effective population size, I mean the number of breedable individuals. If you can see here, this is the definition from Falconer book, from that book, number of individuals that would give rise to the calculated sampling variance or rate of inbreeding if they bred in the manner of the idealized population. Okay, and mu is the mutation rate that is taking place in that generation. So this is how the diversity is existing in a population. We will enter into the topic in the next class. So here you can see that how can you measure the effective population size. This is a condition where that is I have depicted only one condition, but if you go to Falconer you will find many such conditions like a number of individual that changes from generation to generation, if the family size also changes, if there is unequal numbers, if there is selfing, if there is minimal inbreeding, what will happen to the effective population size. But this is the most important one. If there is unequal sex ratio, then effective size can be calculated like this. Say for example you have 100 individuals, 60 are of females and 12 males, breedable males, then how will you calculate? from here you can see that the effective population size is coming somewhere around 40 by this equation. So what it says, as because it is a Wright-Fisher model or a neutral model, and it follows the idealized condition as well. So even though we have 100 individuals, it is equivalent to 20 males and 20 females, because 40 individuals you have to divide it equally, because in that idealized condition you should have equal males and equal females. So in an idealized population, if there had been any idealized population somewhere, then it is equivalent to 20 males and 20 females of that ideal population. And this is our real population, having 100 individuals. So in that way, we have also calculated the number of effective individuals or effective population size of our own herd, the Brindavani cattle. We have calculated it, and I will discuss it later on. Means how the population genetics is being used for our genomic study. So I would like to conclude here. I mean, in the next part, in the next lecture, I will discuss that whether we are still in genetics frame of mind, population genetics, or we have entered slowly into the genomics frame of mind. And after this, I mean, in the next class, in the next lecture, I will discuss about these basic concepts of population genetics. I have touched very little things. I mean, I have not gone to that size, the linkage disequilibrium values, the haplotype blocks, and all those things. But heterozygosity content in that population. But these population genetics parameters, or the diversity parameters, how we can use that, and how that knowledge had helped us in achieving some of the interesting phenomenon of genomics. I mean, we use the genomics information, and with the help of that, and then the population genetics concept, we could easily establish some of the phenomenon that I'm going to discuss in the next class. Thank you very much.